Hi guys. Here is the cardiovascular physiology PowerPoint that you were going to have before respiratory system, but I had made respiratory system for the rest of the classes who are just a bit ahead of you. So this PowerPoint is not very long, but it does have some pretty intense physiology in it. So we're going to go through it pretty slowly, and feel free to go over this recording as many times as you need to understand the physio behind it. And if you still have questions, please email me so that I can set up a Zoom or a FaceTime appointment with you to explain the physio behind the cardiovascular system. Don't forget that the slides that I skipped, they are still fair game for the exam, but they are slides with information that you should already know. I'm just repeating them for you just to kind of make sure we're all on the same page. So the first thing you're going to see is a 4-chamber view. It's an anterior view of the heart, and you see that here and here are smaller chambers, and down here are larger chambers. The 2 smaller chambers are called the atria. The 2 larger chambers are called ventricles. And you see that they work together to have the atria contract and then the ventricles contract. They fill up, and then the atria contract, then the ventricles contract. So they do work together to get both atria contracting at the same time and ventricles. Here is a superior view of somebody's heart. This is the vertebrae, and this is the anterior of their body, probably their thoracic region. In this superior view, you see this really big valve, and it's a pretty large blood vessel as well, and it has 3 flaps to it. And this one is going to be our aortic semilunar valve. Every time the heart pumps, you see that valve go ahead and open. Now, this is a graph with x-axis being the EKG, you see PQRST wave, and the y-axis being different pressures. It's measured in mmHg. What we're going to do is we're going to measure each of these 4 pressure curves and match it up with which part of the EKG it goes along with. Now, to understand this slide, you need to make sure you've gone over the previous cardiovascular PowerPoint, because you have to understand what's happening in each one of these waves. This is something that we already went over. Nobody has emailed me with questions about this, so my assumption is that everyone understands it. So I'm just going to go through as if you have gone over it, you understand it, and you know how to read an EKG. So the P wave is where you have atrial depolarization. So we're going to look over here, and we're going to look for something that has to do with atria. This is aortic pressure, atrial pressure, ventricular pressure, and ventricular volume, which is measured in cc's. So the unit of this guy is going to be a little bit different than the unit for these 3 pressures. For the P wave, this is going to be atrial depolarization. So this is when the atria are going to be contracting. And when the atria contract, it's going to generate a pressure. But do the atria need to contract with a very, very high pressure? No, because the atria are simply pumping the blood right down into the ventricles. So if we look at the atrial pressure line, this arrow points to atrial pressure. It's this solid line that goes up, drops down, goes up, and drops down again. What we want to look at here is this beginning portion. When the P wave hits, that means atrial depolarization happens, which means the atria have contracted, which means that the atrial pressure should be rising. That's going to be this atrial pressure right here. So you see the slight rise from here up there. Sorry guys, that's my dog. This atrial pressure goes up slightly, and that's what causes the contraction. So atrial pressure rose right when the P wave hit. Now the QRS wave, QRS is going to be 2 different things. Number 1, it's going to be ventricular depolarization. So when the ventricles are depolarizing and contracting, at the same time, you're going to have atrial repolarization. So atria are going to be relaxing a little bit. So QRS wave, we're going to go up and match it up with a few different things. The first thing I want to match it up with is ventricular pressure. During a QRS wave, when the ventricles are contracting, what can you expect the ventricular pressure to do? Is pressure going to go up in the ventricles or down in the ventricles when the ventricles contract? Well, pressure in the ventricles should go up when it's contracting. So let's follow ventricular pressure, the dotted line. So ventricular pressure is low, and then it shoots up. And when it shoots up, that's when the QRS wave is happening. So when voltage is traveling through the ventricles, it causes the ventricles to depolarize and contract, causing the ventricular pressure to shoot up. Now, when ventricles contract, remember that the ventricle contracts in order to pump the blood out of them. So the next one we're going to look at here is ventricular volume. The volume of blood in the ventricles is going to drop down during the QRS complex because the volume from the ventricles emptied out, so it went down. Where does blood go from the ventricles? Well, the right ventricle sends blood to the lungs. The left ventricle sends blood out of the aorta. Because of that, we look at aortic pressure now, pressure in the aorta. During the QRS complex, the ventricles contract. The left ventricle pumps blood out of the aorta, so pressure in the aorta is going to rise as well. If you notice, aortic pressure is the solid black line, and it bounces up right at the QRS complex. So right here on the right side, it kind of breaks down everything for you, the PQRST waves. And then I've included here end diastolic volume and end systolic volume. This just means the volume at the end of diastole. Again, diastole and systole are terms you should know. Diastole is ventricular relaxation. And when the ventricles are relaxing, really what's happening is they're filling with blood. So they should have the maximum amount of blood at their end diastolic volume. When the ventricle contracts, shoots all that blood out of it, that's going to be the volume called the end systolic volume, which means the volume of blood at the end of systole, aka ventricular contraction. So again, if you struggle with this, here's the point of all this. Match up P with what's going on in the atrial pressure, and match up the QRS complex, what's happening in the aortic pressure, and ventricular volume. So some of this will be a review for you for how to regulate blood pressure. You can regulate blood pressure using two major systems, the nervous system and the endocrine system. The nervous system, depending on how you stimulate it, can increase or decrease blood pressure in your body. So the nervous system in and of itself, it has receptors for pressure called baroreceptors. Baroreceptors constantly send signals to the brain to keep telling your brain if your blood pressure is too high or too low. Let's say your blood pressure is very high for some reason or another. When blood pressure is too high, the baroreceptors will sense the high pressure and they will send a signal to the autonomic nervous system. Remember, this is the automatic nervous system, sympathetic, parasympathetic, to help release neurotransmitters that are going to cause vasodilation. Now, in terms of you maintaining homeostasis, if you're just relaxing and for some reason your blood pressure goes higher, and that high blood pressure is sensed by a baroreceptor, the signal is sent to your brain, and the brain says, uh-oh, blood pressure went up for some odd reason. How are we going to bring it back down? Well, the blood vessels will vasodilate. If you dilate or make larger your blood vessels, those blood vessels are going to have lower pressure fluid in them. Think of it as a pipe with water running through it. If you make the pipe larger, the water pressure inside is lower. But if you make the pipe skinnier and less in diameter, the pressure of the water is going to be higher in there. So vasodilation means opening up blood vessels by relaxing the smooth muscle, because remember, blood vessels are surrounded with smooth muscle. Now, the opposite thing will happen if your blood pressure is too low. If for some odd reason, as you're just relaxing, sitting down, your blood pressure drops, the baroreceptor will sense it. It'll send a signal to your brain, and the brain will say, hey, blood pressure is way too low. How can I increase blood pressure a little bit? Well, I can cause vasoconstriction by constricting the smooth muscle. And if I vasoconstrict, I'm going to decrease the diameter of the blood vessel, and it's going to increase the pressure of the blood inside of it. So by simply using baroreceptors and allowing for vasoconstriction or vasodilation, I'm able to adjust the blood pressure in my body. This last one here is about the endocrine system, and that's a system of hormones. And one of the big ones I want to just mention to you now, we're not going to go into the physiology of it, is your heart. Your heart is able to sense when the pressure inside of it is too high. And when the heart says, the blood pressure inside of me is too high, I'm being stretched out too much, I don't like it, the heart is going to send off a hormone called ANP. Again, it's called ANP, atrial natriuretic peptide. ANP, the job of this hormone is to go to the kidneys and tell the kidneys, hey, you can go ahead and pee out more water, because if I pee out more water, I'm going to lower my blood volume and therefore lower my blood pressure. So how do cardiac cells contract? Well, in this next physiological mechanism you're going to see a cardiac cell at rest and then one that is in its contraction stage. Now, remember, these are just cardiac muscle cells. These are not the pacemaker cells like the SA or the AV node cells. So when the SA and AV node do depolarize, they send off a voltage signal, and that voltage signal travels through the entire heart from cell to cell very quickly through those gap junctions we call intercalated discs. Now, when that voltage touches each cardiac muscle cell, it's going to take it from a relaxed state to a contracted state. How is that? Well, because there are 4 transporters that work off of voltage changes. One here, we've got one here, this guy there, and then this one here. I'll number them 1, 2, and 4. So 4 transporters. You'll see the same 4 here, but you'll notice that their functions are a bit different. This is when voltage is low, so we'll put voltage is low here. And this is when voltage is high. I also want you to notice that in relaxation state, cytoplasmic calcium is at 1 nanomolar, where in contracted state, cytoplasmic calcium is at 100 nanomolar. Calcium is required for all muscle contractions because calcium binds to troponin to push tropomyosin out of the way, allowing the myosin head to bind and contract. So in a cell at rest, when the voltage is low, that means you don't have a depolarization running through the cell. You have 2 calcium channels here, and both calcium channels are closed. Now, normally calcium is found in high concentrations in the extracellular fluid and in the SR. So when these channels are open, calcium generally wants to leave the SR and go into the cytoplasm, or go from ECF down into ICF. So you'll see that over here in this example. But for now, when voltage is low, the calcium channels are closed. Calcium stays on the outside of the cell or stays stored in the SR. So that's transporter 1 and transporter 4. They're both calcium channels. Now, number 2 here is a sodium-calcium exchanger. Notice that it brings in 3 sodiums for every 1 calcium. This exchanger doesn't require ATP because between calcium and sodium, which one of these is moving down their concentration gradient? It would be the sodium, Because sodium's moving down its gradient, it's able to pull calcium up its gradient and cause calcium to leave the cytoplasm and go back to the extracellular side. Now, why would you want to do this? Well, hey, the cell's in the relaxation phase. When you're relaxed, that means you're not contracting. And if you're not contracting, do you need calcium in the cytoplasm? Probably not. So we're going to actively try to get calcium out by using this antiporter. The last one here, this is called the calcium ATPase because it uses ATP. It is forcing calcium from the cytoplasm into the SR. Why does it use ATP? Well, because you're moving calcium against its concentration gradient into the SR where there's already a lot of calcium. So this will require ATP. I want you to notice this guy here. It's a little membrane protein called phospholamban, and it's found only in the membrane of the SR. And I'm bringing this to your attention because we're going to see how this guy's function changes in the contracted cell. But for now, calcium is pulled into the SR using ATP because it goes against its concentration gradient. Now, all these 4 transporters are going to be in a cell at rest. But once the voltage in that cell goes up, aka it depolarizes, a few things are going to change. Calcium channel 1 and our calcium channel here are going to open. You can call these voltage-gated calcium channels because when voltage is high, they both open, and when they open, because it's a channel, the only direction it can move calcium is down calcium's concentration gradient. So calcium is going to move from the outside into the cytoplasm, and it's going to move from the SR into the cytoplasm as well. Another change that happens is the sodium-calcium exchanger. What do you notice that's different about this one here and that one there? Well, if you said it switched sides, you would be right. Calcium is now getting moved into the cell while sodium is getting pumped out of the cell. Who's going against their concentration gradient now? Sodium. Sodium goes against its gradient without the use of ATP because we're using the downward gradient of the calcium. So this sodium-calcium exchanger changes directions when voltage is high. You can also call this a voltage-gated transporter. Oops. The last one here is again the calcium ATPase, but I want you to notice that this arrow is much smaller than this big arrow. That means that you had more movement of calcium here and less movement of calcium here. What do you notice about the phospholamban? Well, in the relaxed cell, it was pretty far away from the calcium ATPase. But when voltage went up and the cell depolarized, the phospholamban moved across the membrane and this protein bound onto our calcium ATPase. Remember, anytime you bind anything to a transporter, it changes the structure and therefore the function. So instead of bringing in all the calcium it was before, its function has slightly changed and it's only bringing in small amounts of calcium now, which is great because what's the whole point of this depolarized cell? Well, it's to contract. And for this cell to contract, it needs to have as much calcium in the cytoplasm as possible. So we're bringing it in from here, we're bringing it in from here, moving it out of the SR, and we're trying not to store so much from cytoplasm back into SR. You want to keep as much calcium in the cytoplasm as possible. So maximal calcium concentrations here. So again, the transporters are the same, but their function changes dependent upon the voltage of the cell. So I'm gonna have my pen again because I gotta draw something here. This is the outside of the cell, and then we'll extend this membrane, and this is the inside of the cell. So Talking about those voltages, we're going to talk about one specific transporter and voltage changes. The sodium-calcium exchanger. It exchanges 3 sodiums for every 1 calcium, regardless of what direction it's going. Now, how do we decide what direction this sodium-calcium exchanger runs? Well, the voltage decides that. And with the voltage deciding that, we have a threshold here of negative 20. So when voltage of the muscle cell is more negative than negative 20, that means negative 30, negative 40, negative 50, sodium is going to be transported out of the cell and calcium will be transported into the cell. So when you're more negative than negative 20 millivolts, that means you're at rest. You're not being activated, you're not depolarizing. And when you're at rest, again, the goal is to get calcium out of the cell, which is why the direction of the sodium-calcium exchanger is going to be moving sodium into the cell and calcium out of the cell. But what happens when the membrane voltage becomes more positive than negative 20? That means you are finally depolarizing. That cell is going to activate. At this point, you're going to need calcium. So the exchanger changes directions and sodium gets transported out of the cell and calcium now gets transported into the cell. So what you see over here is the example of calcium moving out and sodium moving in. Based off of what you learned in these 2 bullet points, would this be a cell that's active or relaxed? Think about it for a minute. I'll give you a moment. So calcium's moving out, sodium is moving in. Is this going to be a cell that is going to be contracting or relaxed? So if you said relaxed, you would be correct. How do I know that? Well, because when we're relaxed, we don't need calcium in high concentrations in the cell. So we're going to pump it out. At the same time, you're going to pump sodium in. So this is this guy here. So membrane voltage is more negative than negative 20 and we are relaxed. Now, what you didn't see in the last image with the cells is that when we pump sodium in, so far from what you've learned, do you want a sodium concentration building up inside your cell? Well, no, you don't. So what transporter do we have that you've learned about that can take sodium out of the cell? And remember, you're going to definitely be pulling it against its concentration gradient. Well, the sodium ATPase. So in this illustration, it does show you that although sodium gets in, gets right back out. And we pump potassium in. Now, if you don't understand this quite yet, pause the video and go back, rewind it, rewatch the previous slide and this portion of it, because the next thing I'm going to talk about is medications that shift the function of the sodium-calcium antiporter. These medications that I'm going to list are used for people who have heart conditions. Digitalis, Wabane, and digoxin. All 3 of these, they do have the same function, and their function is to lower the transport rate of the sodium-calcium exchanger. So again, this is still the outside, and the other side is still the inside. So normally at rest, you want to be pumping out a lot of calcium out of the cell because you're at rest. But when you take these medications bind to the sodium-calcium exchanger, and they don't shut it down, they just slow it down. So we still pump calcium out, but much less. You're still pumping sodium in, but much less. Now you're wondering, how would this help someone who has a heart condition? Well, if someone has a condition where their heart rate is beating way too fast and each of their heartbeats has a very low contractility. So their heart's beating really fast but really weak. You want to shift that. You want to slow down the heart rate but increase the contractility to make sure their heart doesn't get too tired and it doesn't develop heart disease. So what does this have to do with any of those outcomes? Well, these medications have two roles. One of the roles here, it has nothing to do with this transporter. So let's just cover this one really quick so I can teach you the physio of the second role. The medications like digoxin, they slow a patient's heart rate down because they're able to not completely block electrical impulses, but simply reduce the number of electrical impulses that travel from the AV node down the ventricular septum towards the Purkinje fibers. So if you recall, the direction of movement of voltage is SA node to the AV node to the bundle branches and up and around through the Purkinje fibers. If we can reduce the number of electrical impulses that travel from the AV node to the Purkinje fibers, you'll be able to decrease the person's heart rate. The mechanism of this is not something that we are going to go over in this particular class. The second function, though, the one where you're strengthening the contractions, so you're strengthening the contractility of the heart, that is the mechanism that correlates to this transporter's function. Now remember, the more myosin heads that bind to actin, the stronger the muscle contraction is going to be. The way I'm able to adjust this is by making sure I have high amounts of calcium binding to troponin to expose as many myosin binding sites as I possibly can so I can get as many contraction bridges formed. So how do I do that? Well, if I decrease the amount of calcium leaving the cell, this calcium that's in the cell is going to eventually get stored into the SR. So if I reduce calcium efflux, that means letting calcium out, more calcium stays inside the cytoplasm of the cell, which will eventually get stored into the SR. And if you have more calcium in the SR, that means during your next contraction, that SR is going to release double the amount of calcium that it did before. So just by increasing how much calcium you store in the SR, that will increase your contractility because when more calcium gets released out of the SR, it binds to more troponin to allow more tropomyosin to move out of the way and more myosin heads bind, increasing contractility. So again, it's important to understand the normal function of the sodium-calcium exchanger to then understand how medications change that function. This is just a repeat of the function on the last slide. So a little bit about pacemaker cells. Pacemaker cells are special cells, and the reason is because they have a special channel in them that other cells do not. So pacemaker cells do exactly what the name states. They set the pace of the heart, and they do this at different rates. Pacemaker cells are found in different places of your heart, and they're going to be found in the SA node, the AV node, and the Purkinje branches. SA node, their pacemaker cells function and send off depolarizations at the highest rate of 80 to 100 beats per minute. AV is 40 to 60 beats per minute, and Purkinje fibers are 30 to 40 beats per minute, which means that if the SA node poops out and stops functioning, you're not just going to drop dead. Your AV node will take over. Yes, your heart rate will decrease overall, but it will still beat. This idea that one set of pacemaker cells takes over for another is called override suppression. You're able to override the function of the previous cells with the function of the coming cells. Now, we've seen the graph like this before, and I'm going to kind of go through it right now just a little bit, but in the next slide, we're really going to dive deep into it. Pacemaker cells have a little bit of a higher resting membrane potential, and that's so that they can reach threshold more quickly. You want to make it as easy as possible for your heart to actually beat. So your heart's going to slowly increase the voltage until it meets the threshold. At threshold, it shoots up an action potential, aka your voltage, and then it has to come back down to rest so it can do the next one. Each firing that the heart or that the SA node does is going to shoot off voltage signals down to the AV node. So, how does this work? Let's get my pen and show you this. So, this graph up here overlaps with this graph down here. Let's go over this one first. The voltage of the resting membrane potential of a pacemaker cell is around -65, a little bit higher than normal cells. The voltage slowly increases and then it shoots off an action potential and comes back down to resting. But if you notice that this line where the resting membrane potential is, it kind of surpasses that. And what is it called when the voltage gets lower than the resting voltage? It's called hyperpolarization, and there is a reason for that. So pacemaker cells, increase voltage, shoot off an action potential, relax. Slowly increase, shoot off an action potential, then relax. During each one of these increments, and I'm going to circle them, let's see, that one, and then this one coming down here. I'm going to call it 1, 2. This is harder than you think to do on a laptop because I don't have an iPad, which I probably should invest in too. So 1, 2, and 3, the 3 phases of pacemaker cells. They correlate with 3 ion movements. We have 1, and I'm going to number these for you because they match up with those guys. 2 and 3. Well, that was a weird 3. Okay, so these 3 are 3 different ion movements. They're written over here. ICA is calcium movement, IK is potassium movement, and IF, I know this is really weird that they named it IF, is sodium movement. They named it IF because before they found out that they were sodium channels, they were just funny channels. So scientists legit name this the funny current. So what we have here is inward movement and outward movement of ions into or out of the pacemaker cell. If the line or the curve is underneath this flat line, that means you have an inward movement. So sodium and calcium are going to be moving into the pacemaker cell. Above it is outward movement. The only ion we have is potassium that's going to be moving out of the pacemaker cell. Now, let's start with this first part, and I'm going to kind of draw a line moving down here and here. Oops. Here. So this slow rise of voltage until we get to this threshold point is caused by this dotted line. See how it's matched up on top of each other? What is this dotted line? Well, it's labeled IF, and IF is an inward sodium current. So, because of an inward sodium current, think about what charge sodium has. It's a positive charge. As sodium trickles into the pacemaker cell with its positive charge, what's going to happen to the overall voltage of the pacemaker cell? Well, it's going to slowly rise up. This is the channel that makes pacemaker cells so special. I'm going to write down a word here. Let's see if I can do this on my mouse. Leaky. Man, it looks like a toddler wrote this. Leaky. These are leaky sodium channels. There are no other cells in your heart that have leaky sodium channels other than pacemaker cells. Leaky sodium channels literally do what the name says, and they allow sodium to leak into the cell. Very slowly though. That's why there's a long slope up. So sodium leaks in, leaks in, leaks in, leaks in, and then boom, you reach the threshold. The minute you reach the threshold, your next channel is going to open, number 2. What channel's number 2? Oh, there it is, the calcium channel. And is calcium going to be moving into the cell or out of the cell? Well, the curve is below the line. That's an inward movement. It's moving into the cell. So when you reach threshold, calcium, look at how big that inward current is. This is a huge inward influx of calcium. And what charge does calcium have on it? A 2+ charge. With all of that calcium, with all of its positive charge rushing into the cell, you can expect that the voltage of that cell is going to shoot up with a very steep slope. That, my friends, is the voltage shoot off. That is our action potential. Now, when we get to the top of this curve, you do have a maximum voltage that you reach, at which point the calcium channels are going to shut and calcium influx will go down. And at the same point here, if you notice it matches up right at the tip of this point, number 3 happens. What is number 3? It's potassium. What is potassium doing? Is it moving into the cell or out of the cell? Well, this curve is above the line, so out of the cell. Because potassium has a positive charge, it has a +1 charge. When you have potassiums now rushing out of the cell, that means positive charge is rushing out of the cell. And if positive charge rushes out of the cell, the voltage of that cell is going to start going down. And because potassium channels are kind of lazy, they take a long time to close. So even once you reach the resting potential and all the potassium channels should shut, potassium channels need a little bit of time to actually close all their doors, which is why we hyperpolarize. So I'm going to circle this word here. It is important. So again, this is the voltage change of the pacemaker cell, and this graph is the movement of sodium, calcium, and potassium, and they match up with each of the points, or I guess each of the curves from the voltage graph. Now, to kind of show you what different medications do. There are medications that speed up your heart rate. There are medications that slow down your heart rate. In these graphs, you're going to see that in the solid black line is a normal heart rate. We got 3 action potentials here. And again, 3 action potentials here. This top graph has to do with sympathetic innervation. The bottom graph, parasympathetic innervation. So let's say from a normal heart rate, of which you already know the physiology for, you elicit the sympathetic response. Let's say something scared you and now you're panicking a little bit. When you get scared, your body releases norepinephrine. You know this, epinephrine, norepinephrine gets secreted in the sympathetic panic response. Norepinephrine is going to bind to beta receptors. You also learned this in, I think, the last week that we had together. Norepinephrine binds to beta-1 receptors that are found in the SA node. When the norepinephrine binds to the receptor in the SA node, it causes an increase in C-amp. C-amp, remember, is the internal second messenger of cells. Now, this is the part that's important. We increased C-amp in the cells, and an increase of C-amp has 2 major effects. Number 1, the more C-amp you have, the more leaky your sodium channels are. So let me go back a slide. Remember, this rose up because of leaky influx of sodium. If I let in a lot more sodium faster, am I going to reach threshold faster or slower? The answer is faster. The more sodium that gets in, the faster I'm going to reach my threshold because my voltage is going to go up. So, C-amp increases the sodium influx current, which means we're going to reach threshold faster, which means we're going to depolarize at a faster rate. The second thing that high C-amp does is that high C-amp, if you recall from, I think it was I know it was the first exam, we know that C-amp can activate kinases. What is the job of a kinase? It is to phosphorylate things. So, as C-amp goes up, the amount of protein kinases you have go up. And with more protein kinases, we're going to be able to do a lot more phosphorylation, specifically of calcium channels. When you phosphorylate calcium channels, it increases their ability to function. So you're going to have more calcium pump activity, which causes more calcium movement at a faster rate. And that means we're able to store calcium at the end of each contraction cycle a lot faster. So here's the gist of this. The faster I can store calcium back in my SR, the faster I can get to my next contraction cycle. Because remember, I'm only at rest when the calcium is stored. So again, the faster I can pump calcium into the SR or out of the cell, the faster my body can get my heart to the next contraction cycle. The parasympathetic innervation, when you are relaxed, your brain knows you're relaxed, so it sends a signal through the vagus nerve, and the vagus nerve releases acetylcholine onto the SA node. Acetylcholine binds to acetylcholine receptors. You saw these guys when we talked about neuromuscular junctions. So, what's the significance of this? When ACH binds to acetylcholine receptor, it does the complete opposite of number 1. It lowers the sodium leaky channel current. It lets smaller concentrations of sodium into the cell, which means it's going to take longer to reach threshold and conduct each action potential. The second thing it does is a little bit different than the sympathetic nervous system example. When acetylcholine binds to acetylcholine receptor, it's going to increase potassium conductance. Now, let's go back a slide. Potassium conductance, the outward movement of potassium, causes this to repolarize back down. If I let more than normal potassium out of a cell, we're going to get even more hyperpolarized. We're going to get lower down. And if we got lower down, let's see, hyperpolarize even farther down, is it going to take us a longer time or a shorter time to reach the next action potential? Well, it's going to take us longer because we're even more negative now. So it takes more time to reach the threshold and shoot off an action potential. So because acetylcholine bound to the acetylcholine receptor, we were able to increase potassium conductance to allow more potassium to leave the cell, which caused the voltage of the cell to drop past down its resting membrane potential to further hyperpolarize, which means we're further away from the threshold for the next contraction, and we're lowering the heart rate at that point. So, I know this is a difficult lecture, and I'm so sorry that we were not able to do it together, my Monday, Wednesday class. And if you need any help on this, or you need me to go through all of the physio slides with you, please just email me and let me know. But the key thing here is that you have to understand the normal function of muscle cells you have to understand the normal function of our pacemaker cells here, and you put them together to understand this slide here. The last thing we're going to cover is two medications that you may have heard of. They use them a lot in the emergency room. One is called atropine, the other is called propranolol. So they did an experiment and they gave patients a dose of atropine, and then they gave the same patients a dose of propranolol. This is going to be the blue over here. And the patients that got atropine first, they saw that atropine increased the heart rate. Then propranolol did the opposite and decreased it. For people that got atropine, for people that got propranolol first, it lowered the heart rate. And then when they got atropine, it definitely increased the heart rate. Now, what is the physiology of these drugs? Well, atropine increases the heart rate by blocking acetylcholine receptors. If you block acetylcholine receptors, what is not able to bind to them anymore? Well, acetylcholine can't bind to them anymore. And if you can't bind acetylcholine, that means your heart rate is not going to stay in the parasympathetic phase, and it's actually going to go up to sympathetic-like characteristics. Propranolol lowers the heart rate. What receptor do you think this guy's blocking? If we're lowering the heart rate, we're going to be blocking beta receptors. Beta receptors normally bind to things like epinephrine, norepinephrine, and adrenaline, which is the street name for those two. If you block the binding of norepinephrine, you're going to allow the heart rate to slow down because you're basically blocking the sympathetic innervation. So again, to understand this slide, you got to first understand this slide and notice that there's a beta receptor here and an acetylcholine receptor here. This PowerPoint does very much build on itself. So be sure that if you're confused, go back to previous slides, make sure you understand them one slide at a time and build it for yourself before moving to the next slides. Again, if you need any help with this, please go ahead and email me. I will be posting this for all sections, and I hope you guys all have a great weekend and are staying healthy and happy. Talk to you soon.